Hello everybody, welcome to all to my presentation. My name is Emran. I'm a new faculty, Department of Plant Pathology, University of Georgia Tipton campus. My today's title, Molecular Characterization and Sensitivity to Quinone Outside Inhibitor QoI Fungicides of Aspergillus flavus isolated from peanut seed in Georgia. At first, I would like to give you an outline of my today's talk. First, I would like to mention a brief overview of Aspergillus flavus problem in peanut seed production, and then I'll focus molecular characterization of Aspergillus flavus isolated from peanut seed in Georgia. In this regard, 2 points would be mentioned. One is how we did molecular ID, and secondly, how we detect and quantify aflatoxin production status of this isolate. And thirdly, I'd like to talk on in vitro sensitivity of Aspergillus flavus isolated to the quinone outside inhibitor QoI fungicide. As you all know, peanut seed are affected by several Aspergillus species like Aspergillus flavus, Niger, Parasitica. So among them, Aspergillus flavus is one of the important pathogenic Aspergillus species that affect peanut during storage condition, and it also has an importance regarding aflatoxin production, which is potent human livestock carcinogen. So let's talk about your current issue. What's our current issue? The current issue is this year we have seen a higher incidence of Aspergillus species infection, probably Aspergillus flavus, and then the regular seed treatment is failed to control this pathogen. As a result, we have seen very low seed germination, around as expected level is 90% or above, but it's going down to 40-50%. So our research is focusing to seek answer whatever the isolate we collected from peanut seed, are they solely belongs to Aspergillus flavus? Since this pathogen is a aflatoxin production pathogen, so we would like to see what's their level of aflatoxin production status. And finally, we also would like to know what's their current level of sensitivity against QoI fungicide Azoxystrobin. For this trial, we have collected our isolate from 6 different commercial seed lots in southwest Georgia. Total 76 isolate were collected and isolated and purified on potato dextrose agar. Molecular characterization, alpha-toxin status, and QoI sensitivity analysis were performed at the Plant Molecular Diagnostic Lab, University of Georgia Tipton campus. Characterization of this isolate were conducted using phenotypic characterization and molecular characterization. For phenotypic characterization, as you know, based on colony morphology, color, patterns, so we can easily distinguish them as a Aspergillus flavus. For the molecular characterization, initially we use universal Aspergillus primer that can amplify all kinds of Aspergillus species to going down to be more specific. Later on, we use Aspergillus flavus specific PCR by targeting 18S rDNA, and as you see, we have obtained the expected size of band that can confirm as a Aspergillus flavus. Further, we also conducted a sequencing of this isolate, and we confirm all those species belongs to Aspergillus flavus. Later, we have conducted phylogenetic analysis to see the genetic variation among Aspergillus flavus isolate collected from Georgia. We have done first the comparison among those isolate each other collected from Georgia, and we have seen their ID is very much similar. And then we did comparison with other Aspergillus flavus isolate that are submitted in gene bank, and as well we also compare with other Aspergillus species. As you've seen from this tree, our Aspergillus flavus isolate is quite distinguished than other Aspergillus flavus isolate based on this beta tubulin gene. The next, we move on to see the aflatoxin status of this isolate. Here we try to detect the 4 gene those are mostly involved in aflatoxin biosynthesis pathway, as you know, NOR1, WRD1, OMT, and AF1R. All 4 genes we have successfully detected most of our isolate. In contrast, we did not see any amplification in Aflagard. There is a non-alpha toxicogenic isolate. To quantify those genes more accurately, further we have conducted real-time PCR technique to determine the expression level of all 4 genes that are related to biosynthesis pathway. As you can see, here's the non-alpha toxicon isolate expression compared to all other isolate from Georgia. Their expression definitely higher, and that can tell us in our Georgia isolate has indeed has alpha toxin expression. So from my first part, I'd like to summarize. So all the isolate we collected from Georgia peanut seedlers are confirmed as Aspergillus flavus. Among Georgia's isolate, we did not see genetic variation. However, we do see this variability among all other Aspergillus flavus isolates submitted in gene bank, and also as well as some other Aspergillus species. So all 4 important genes, those are involved in alpha toxin biosynthesis pathway, we analyzed, and we found higher expression in compared to non-alpha toxicogenic isolate Aflagard. Now I'd like to talk my second part of my research, is sensitivity of Aspergillus isolated to the QoI of fungicide. So in this experiment, we use all 76 isolate collected from different seedlot in Georgia, and we have determined the EC50 value based on mycelial growth inhibition assay using different concentration, 0.1, 1, 10, and 100 microgram per ml. So we use potato destruct PDA media, and the spore suspension we use for this experiment is 1 times 10 to the power 5 spores per ml. So as you can see this slide, there is a representative sample what we did for this in vitro sensitivity analysis. Most of our isolate has shown the very reduced efficacy against this pathogen. So as you can see, even some pathogen or some isolate grow even 100 ppm, and definitely we have seen some isolate they cannot grow on higher concentration. Here's we determine the EC50 value distribution among those isolate. As you can see, we have seen in this graph there is a 2 range of EC50 value, 0.1 to 1, 1 to 10, and more than 10. So most of our isolate belongs to more than 10 ppm the EC50 value. However, some others have the quite similar number to these other 2 ranges. So mean EC50 value for our all isolate is 9.07, and this range is quite very big, like widen 0.122 all the way to 97.22. So definitely the variation factor goes higher. So based on this result, at least we can see the reduced efficacy of QoI fungicide against these Aspergillus flavus isolate. The next question comes, why was that? So is that reduced efficacy is related to any kind of mutation that is already reported in different articles? To answer this question, we have done sequence analysis on cytochrome B gene for all other isolate, and indeed we see these 2 mutation that has already reported for fungicide resistant phenotype. One is F129L and G143A. Most of our isolate have indeed a mutation, either one or another. So 37% isolate we have seen G143A mutation, and 16% isolate we have seen F129L mutation. Both are together more than 50% isolate have indeed this mutation. So This result showed a correlation that indeed this mutation might have effect for this reduced efficacy of this fungicide. Later we have tried to determine the frequency of QoI mutation in regarding the resistant phenotype. So as you can see, the non-mutated isolate, the EC50 value is so low. However, G143A, the average EC50 value really high compared to F129L mutation, the EC50 value around 50. So when we see how they look in distribution, their EC50 value, you will see when it comes to the non-mutated isolate, the most of the isolate belongs to less than 1 EC50 value. Very few of them has 1 or a little more EC50. However, when you talk about G143A mutation isolate, those isolate EC50 value, most of them more than 10. Some of them has close to 10. For F29 isolate, you will see definitely they have some isolate has more than 10 EC50 value, but still some of them are quite same number as the close to 1 to 10 EC50 value. Based on this trial and our experience, we determine a discriminatory dose that could be applicable as a discriminatory dose against this fungus. We determined 2.5 ppm could be the best. So we used, and further experiment, we used 2.5 ppm dose and try to grow all our isolate, and we determine what's their resistant phenotype looks like. As you see this slide, we have after we grow, we have seen different types of grow. Some are isolate did not grow at all, some grow nicely, some has effect on their growth. Based on their growth pattern, we have determined their resistant phenotype, especially the relative growth inhibition calculation. So we consider, if we see 0 to 14% relative growth inhibition, we consider it as resistant, highly resistant isolate. If we see 15 to 50% growth inhibition, then we consider it moderately resistant. And if we see 51 to 75% growth inhibition, we consider it reduced sensitive isolate. And finally, if we see 76 to 100% growth inhibition, then we consider it sensitive isolate. Based on this parameter, we have seen our G143 mutated isolate are mostly highly resistant. Yes, indeed, they have some moderately resistant. But in case of F129L mutation, they're almost 50-50. They are highly resistant, moderately resistant, and some are reduced sensitive isolate. Without mutation, we do see a very slight reduced sensitivity, few isolate, but most of the isolate actually sensitive. The summary of my part 2, we have determined this QoI resistant phenotype confirmed based on the phenotypic characterization like EC50 value of Aspergillus flavus isolate from peanut seed in Georgia. A high percentage of our Aspergillus population with known resistant mutation like G143A and F129L. So finally, I would like to give a suggestion. So based on our research, for effective control of these diseases, grower need to focus on alternative to QoIs. Finally, I would like to thank all the personnel involved in this research, especially Elena Bennett, Jessica Bell, Brian Jordan, Owen Harrison, and also the funding agency, Georgia State Development Commission. Thank you all for your patient hearing. Thank you.